this session, we are going to continue from the previous video. So let's try it. Macrophages are activated by a variety of stimuli in the course of an immune response. Phagocytosis of particulate antigens or contact with receptors that sense molecules present in microbial pathogens often serve as an initial activating stimulus. However, macrophages' activity can be further enhanced by cytokines secreted by activated T cells and by mediators of the inflammatory response. Activated macrophages are more effective than resting ones in eliminating potential pathogens for several reasons. They exhibit greater phagocytic activity, an increased ability to kill ingested microbes, increased secretion of inflammatory mediators, and an increased ability to activate T cells. In addition, activated macrophages, but not resting ones, secrete various cytotoxic proteins that help them eliminate a broad range of targets, including virus-infected cells, tumor cells, and intracellular bacteria. More will be said about this later. So, activated macrophages also express higher levels of class 2 MHC molecules, allowing them to function more effectively as antigen-presenting cells of Th cells. Thus, macrophages and Th cells facilitate each other's activation during the immune response. The next topic, phagocytosis, is followed by digestion and presentation of antigens. Macrophages are capable of ingesting and digesting exogenous antigens such as whole microorganisms and insoluble particles, and endogenous matter such as injured or dead host cells, cellular debris, and activated clotting factors. Phagocytosis is initiated by the adherence of the antigen to the macrophage cell membrane. Complex antigens such as whole bacterial cells or viral particles tend to adhere well and are readily phagocytosed. Isolated proteins and encapsulated bacteria tend to adhere poorly and are less readily phagocytosed. Adherence induces membrane protrusions called pseudopodia to extend around the attached material. Fusion of the pseudopodia encloses the material within a membrane-bound structure called a phagosome, which enters the endocytic processing pathway. In this pathway, a phagosome moves toward the cell interior, where it fuses with the lysosome to form a phagolysosome. So the lysosomes contain a variety of hydrolytic enzymes that digest the ingested material. The ingested contents of the phagocytosome are then eliminated in a process called exocytosis. And the macrophage membrane has receptors for certain class of antibody. If an antigen, a bacterium, is coated with the appropriate antibody, the complex of antigen and antibody binds to antibody receptors on the macrophage membrane more readily than antigen alone, and phagocytosis is enhanced. In one study, for example, the rate of phagocytosis of an antigen was 4,000-fold higher in the presence of specific antibody to the antigen than in its absence. This antibody functions as an opsonin molecule that binds to both antigen and phagocyte, thereby enhancing phagocytosis. The process by which opsonins render particulate antigens more susceptible to phagocytosis is called opsonization. Then, although most of the antigen ingested by macrophages is degraded and eliminated, experiments with radio-labeled antigens have demonstrated the presence of antigen peptides on the macrophage membrane. Phagocytosed antigen is digested within the endocytic processing pathway into peptides that associate with class 2 MHC molecules. These peptide class 2 MHC complexes then move to the macrophage membrane. This processing and presentation of antigen is critical to Th cell activation, a central event in the development of both humoral and cell-mediated immune response. Then granulocytic cells. The granulocytes are classified as neutrophils, eosinophil, and basophil on the basis of cellular morphology and cytoplasmic staining characteristics. The neutrophil has a multi-lobed nucleus and a granulated cytoplasm that stains with both acid and basic dyes. It is often called a polymorphonuclear leukocyte for its multi-lobed nucleus. The eosinophil has a bilobed nucleus and a granulated cytoplasm that stains with the acid dye eosin red, hence its name. The basophil has a lobed nucleus and heavily granulated cytoplasm that stains with the basic dye methylene blue. Both neutrophils and eosinophils are phagocytic, whereas basophils are not. Neutrophils, which constitute 50% to 70% of the circulating white blood cells, are much more numerous than eosinophils or basophils. Neutrophils. Neutrophils are produced by hematopoiesis in the bone marrow. They are released into the peripheral blood and circulate for 7 to 10 hours before migrating into the tissues, where they have a lifespan of only a few days. In response to many types of infections, the bone marrow releases more than the usual number of neutrophils, and these cells generally are the first to arrive at the site of inflammation. The resulting transient increase in the number of circulating neutrophils, called leukocytosis, is used medically as an indication of infection. The movement of circulating neutrophils into tissues, called extravasation, takes several steps. The cell first adheres to the vascular endothelium, then penetrates the gap between the adjacent endothelial cells lining the vessel wall, and finally penetrates the vascular basement membrane, moving out into the tissue spaces. This process is fully discussed. So, a number of substances generated in an inflammation reaction serve as a chemotactic factor. Chemotactic factor that promote accumulation of neutrophils at an inflammatory site. Among these chemotactic factors are some of the components of the blood clotting system, and several cytokines secreted by activated T cells and macrophages. Like macrophages, neutrophils are phagocytes. Phagocytosis by neutrophils is similar to that described for macrophages, except that the lytic enzymes and bactericidal substances in neutrophils are contained within primary and secondary granules. The larger, denser primary granules are a type of lysosome containing peroxidase, lysozyme, and various hydrolytic enzymes. The smaller secondary granules contain collagenase, leptoferrin, and lysozyme. Both primary and secondary granules fuse with phagosome, whose contents are then digested and eliminated. So, neutrophils generate a variety of antimicrobial agents, which will be told later. And eosinophils, like neutrophils, are motile phagocytic cells that can migrate from the blood into the tissue spaces. Their phagocytic role is significantly less important than that of neutrophils, and it is thought that they play a role in defense against parasitic organisms by secreting the contents of eosinophilic granules, which may damage the parasite membrane. Then basophils are non-phagocytic granules that arise by hematopoiesis and function by releasing pharmacologically active substances from their cytoplasmic granules. These substances play major roles in certain allergic responses. Mast cell precursors, which are formed in the bone marrow by hematopoiesis, are released into blood as undifferentiated cells. They do not differentiate until they leave the blood and enter the tissues. Mast cells can be found in a wide variety of tissue, including the skin, connective tissue of various organs, and mucosal epithelial tissue of the respiratory, genital, urinary, and digestive tracts. Like circulating basophils, these cells have large number of cytoplasmic granules that contain histamine and other pharmacologically active substances. Mast cells play an important role in the development of allergy. Then dendritic cells, identified in 1868 during a close anatomical study of skin by Paul Langerhans. Dendritic cells were the first cells of the immune system discovered. The dendritic cell acquired its name because it's covered with long membranous extensions that resemble the dendrites of nerve cells. There are many types of dendritic cells, and at least 4 major categories are recognized: Langerhans disease, interstitial disease, monocyte derived disease, and plasmacytoid derived disease. Each arrive from hematopoietic stem cells via different pathways and in different locations. Langerhans disease are in epidermal layers of skin, and interstitial disease are present in the interstitial spaces of virtually all organs except the brain. As indicated by their name, monocyte derived disease arrive from DC in the sense dendrocytes. Dendritic cells are in epidermal layers of skin and present in interstitial spaces of virtually all organs except the brain. As indicated by their name, monocyte derived disease arrive from monocytes that have migrated from the brainstem into tissues From the tissues, they can move through lymph to lymph nodes or cross back into the bloodstream and use it as an avenue of transport to lymphoid tissue. So, plasmacytoid derived disease arise from plasmacytoid cells. They play roles in innate immune defense and as antigen presenting cells, although there are important differences in the functions and phenotypes of different varieties of disease. They all share display of class 1 and 2 MHC molecules, and the B7 family of co-stimulatory molecules CD8 and CD86 are present. Dendritic cells also have CD40, a molecule that can influence T cell behavior by interaction with the complementary ligand on T cells. Dendritic cells are versatile, occurring in many forms and performing the distinct functions of antigen capture in one location and antigen presentation in another. Outside lymph nodes, immature forms of these cells monitor the body for signs of invasion by pathogens and capture intruding or foreign antigens. They then migrate to lymph nodes where they present the antigen to T cells. Then, when acting as sentinels in the periphery, immature dendritic cells take up on their cargo of antigen in three ways. They engulf it by phagocytosis, internalize it by receptor-mediated endocytosis, or imbibe it by pinocytosis. Indeed, immature dendritic cells pinocytose fluid volumes of 1000 to 1500 m³ per hour, a volume that rivals that of the cell itself. Through a process of maturation, they shift from an antigen-capturing phenotype to one that suppresses presentation of antigen to T cells. In making the transition, some attributes are lost and others are gained. Lost is capacity for phagocytosis and large-scale pinocytosis. Expression of class 2 MHC increase, which is necessary to present antigen Th cells, and there is an increase in the production of increase of co-stimulatory molecules that are essential for the activation of naive T cells. On maturation, dendritic cell abundant residency in peripheral tissue enter the blood of lymphatic circulation and migrate to regions of the lymphoid organ where T cells reside and present antigen. Then follicular dendritic cells. Follicular dendritic cells do not arise in bone marrow and have completely different functions from those described for the dendritic cell discussed below. Then follicular dendritic cells do not express class 2 MHC molecule and therefore do not function as antigen presenting cells for Th cell activation. These dendritic cells were named for their executive location in organized structures of the lymph node called the lymph follicles, which are rich in B cell. Although they do not express class 2 MHC molecule, follicular dendritic cells do express high levels of membrane receptors for antibody, which allow efficient binding of antigen-antibody complexes. Then organs of immune system. A number of morphological and functionally diverse organs and tissues contribute to the development of immune responses. These organs can be distinguished by function as the primary and secondary lymphoid organs. The thymus and bone marrow are the primary or central lymphoid organs, where maturation of lymphocytes take place. The secondary or peripheral lymphoid organs include lymph nodes and spleen and various mucosa-associated lymphoid tissue, MALT, and as gut-associated lymphoid tissue, GALT. These organs provide sites for mature lymphocyte to interact with the antigen. Once mature lymphocytes have been generated in the primary lymphoid organs, they circulate in the blood and lymphatic system, a network of vessels that collect fluid that has escaped into the tissue from capillaries of the circulatory system. Ultimately, this encapped fluid is returned to the blood. Then primary lymphoid organs. Immature lymphocytes generated in hematopoiesis mature and become committed to particular antigen specificity within the primary lymphoid organs only after a lymphocyte has matured within a primary lymphoid organ is the cell immunocompetent, capable of mounting an immune response. T cells rise in the bone marrow and develop in the thymus. In many mammals, humans and mice, for example, T cells originate in bone marrow. Thymus. The thymus, site of T cell development and maturation, is a flat, bilobed organ situated above the heart. Each lobe is surrounded by a capsule and divided into lobules, which are separated from each other separated from each other. Next topic we will be learning with the help of next video.